Thank you for joining us today. My name is Udit Batra, coming to you today from Waters' Immerse Innovation and Research Lab in Cambridge, Massachusetts. I'm proud to represent all my colleagues from Waters who are behind the innovative solution that we are introducing here today. Today, if you're tuning in live, I want you to know that there are dozens of experts from Waters who are standing by to interact with you in our Waters virtual venue. At the conclusion of my remarks, we will feature a 30-minute scientist panel that you won't want to miss. Following the panel, we'll open up the entire Waters virtual venue for product demonstrations, expert Q&A, and breakouts. Okay, let's get started. If you're watching this presentation, it is likely that separation science is a domain you know quite well. Whether you work in biopharma with large or small molecules, food research, food safety, or biomedical research, you are likely using UPLCs and various chemistries or columns within your lab to characterize and define compounds. If your applications include oligonucleotides, bisphosphonates, lipids, or organic acids, for example, then I know of a problem that you all have in common. If you work with one of these or similar metal-binding compounds, then you are forced to deal with the dreaded steps involved in passivation. Essentially, preconditioning your columns and your instruments, sometimes spending hours and precious amounts of sample material to run your analysis, only to end up unsure of your results. This common problem is known as non-specific adsorption, and it is an analytical challenge that cuts across disciplines and applications, in particular, molecules that carry negative charges and can bind to metals. If you work with these compounds, you understand these challenges. Think about how much time and precious sample material you waste in passivating your instruments and columns. We know customers who can take several days and thousands of dollars in sample material to passivate their LC system so they can get a reliable analysis. How many of you are in a similar situation? Well, today that stops. Today, we at Waters have a solution. In the 17 years since Waters introduced the first UPLC, we have wrestled with these same challenges. And we have also seen that your needs as separation scientists have evolved quite a bit, given the increasing complexity of molecules, the need for faster access to better quality data, and the need to extract expert quality data from analysts who are not experts. Our team at Waters has heard you. We have leveraged the decades of separation experience, know-how, along with combined efforts of our material scientists, chemists, and engineers to bring about the new Acuity Premier solution that is here beside me. The key innovation throughout the system and columns are our max peak high-performance surfaces. The system itself has been designed to take full advantage of new column chemistries, providing an end-to-end inert surface flow path that virtually eliminates the need for time-consuming and costly passivation. The Acuity Premier instrument and columns are designed to work together to give you more reliable data about the complex compounds you're analyzing. They typically result in 10 to 100 times higher sensitivity, providing you with the new ability to detect and measure low-level analytes that might otherwise go undetected. It offers greater reproducibility for separations prone to adsorptive losses, meaning less rework or troubleshooting and more confidence in results. It delivers better quality data in less time without wasting precious sample material on passivation. That, in turn, provides greater confidence in every result so you can make critical decisions much faster. We invite you to stay with us to learn more about the Acuity Premier solution from our panel of scientists who have tested these products with a variety of metal-binding compounds. We also invite you to explore the Waters virtual venue that we have designed for you to connect with us and learn more about the new Acuity Premier solution. We firmly believe Acuity Premier will redefine the value that separation science delivers in your lab by helping overcome some of the most complex chromatography challenges. Thank you again for your interest, and with that, I will hand you over to Matthew Lauber and our scientists panel. Matt.
Thank you, Udit. This is indeed a very exciting day for analytical chemistry. My name is Matt Lauber, and I'm a lead scientist at Waters, and I have the pleasure of hosting a panel discussion and special interview for everyone today. And it's on something near and dear to me, given that I'm one of the primary contributors and inventors of the technology. We are introducing this Acuity Premier system that with Premier columns is bringing new inventions to the world for another level of performance in LC. We've seen benefits for just about every performance attribute you can think of. Quite easily, one could say that this is a time when and where separation science is being redefined. You might be intrigued by this and the ideas that you will soon be hearing. We want you to know, just like Udit was saying, that you can be part of the discussion too. While the conversation unfolds, we'll be encouraging you to send in questions using our on-screen tools. For today, we've invited several scientific leaders to join us for a discussion about this new LC technology. I want to jump straight ahead to hearing from one of them, Professor Ian Wilson. Professor Wilson is the chair of drug metabolism and molecular toxicology at Imperial College London. He has been at the pioneering edge of analytical sciences for over 30 years and has unique insights brought on from the fact that he's spent time in the pharma industry as well as academia. Welcome, Professor Wilson. We've been alluding to quite a few things already about the new Acuity Premier system, but we'd all really love to hear your thoughts on a simple statement: separation science being redefined. What does that mean to you?
Well, I've been in separation science for rather longer than I can actually remember. So I started off in 1976 with microbonder pack. And if you remember those columns, then you've probably been around for too long, but they were 10 micron, irregular particles, 4.6 millimeter diameter columns, and they were 30 centimeters long. So you could conduct a small orchestra with them if you had one in the spare time. And it took quite a long time to move from 10 micron to 5, and then to 3, and to get the particle sizes down. And all these were sort of gentle changes, very welcome at the time. And I can remember just how difficult it was to get down to 2 millimeter columns. And it was actually quite tough. And then 28 years after I started, 2004, we ended up with what I think is the current state of the art until today anyway, which was just UPLC, or just UPLC. How can I say that? I remember seeing my first UPLC separations and thinking I'd been born again as a gas chromatographer. The peaks were so numerous and so sharp, and that was wonderful. And of course, everybody thinks that the heart of the UPLC system, well, I suppose it is true, is the column and the 2 micron particles in it. But the important thing to remember is that it was a system, because, 2 micron columns had been around for a while. Nobody's been able to pump them because they hadn't got the pumps. They hadn't got the system. And that's what Waters introduced then, a system that was engineered to deal with the very high pressures that were required to get the best out of those 2 micron columns. And now here we are in 2021, and there's another system coming along, which in its own way, I think, will be almost as important as UPLC. And the pleasing thing is, if you plot the points on the curve, 1976 to 2004, and then 2004 to 2021, the interval between really big jumps is decreasing, which as I approach the end of my career means I might get another big jump in before it's finished. So 2021, a system that will deal with many of the problems that UPLC merely brought into sharp focus by allowing you to get bad results even quicker. So I'll leave you with that as a thought.
Thank you so much, Ian. That's a very, very nice way to set the stage for our panel discussion. As mentioned, we have a couple other scientific leaders joining us, and these are scientists that Waters has the privilege to call their own, Dr. Aaron Chambers and Dr. Diane Deal. Aaron and Diane, allow me to continue with this question to get us started. Aaron, first, would you mind introducing yourself and giving us a glimpse into your thoughts on separation science being redefined?
Sure. Thank you, Matt. So I'm Aaron Chambers. I run our chemistry organization. So that means it includes the columns that Ian talked about, sample prep, automation, and kits. And, Matt, this idea of separation science redefined, I'm smiling because that's exactly what it is. We have been waiting a long time for this. The pride, the excitement amongst the teams, it's palpable. For me, the first thing that comes to my mind is how much time and frustration I would have saved in my 27 years in the lab if we'd had something like this sooner. We're talking about simpler, faster method development, out of the box performance, seeing things we couldn't see, and the sensitivity. If you look at the application work Diane's group has done, it's incredibly compelling. And I can tell you firsthand how impactful this is. I've had the pleasure to work in drug metabolism, discovery, pesticide analysis, and more recently, a real focus on large molecule quantification. I recall, if you permit me a quick story here, a recent oligobioanalysis project. It was a 25-mer, a phosphorothioated oligo. And I could not get reproducible peaks, nor even a peak at all from injection to injection. So I tried some of the classic things. This will sound familiar to many of you. New columns, old columns, equilibrating longer, injecting higher and higher concentrations. Many of you recognize that drill there. And a day and a half later, things finally stabilized. But by this time, I was worried about carryover and how long my system would stay in this condition. And as I look back, this is a classic example of a widespread problem. We recently reran this same assay on the Premier system with a 12x increase in sensitivity and out-of-the-box performance. I am convinced I would have saved days or even weeks of method development time with these new solutions. And fundamentally, good chromatography is as much about preventing interactions you don't want as it is about the way we typically think about it, which is creating the ones you do. So the underlying problem we're talking about here is non-specific adsorption. That's one of those interactions we don't want. And it manifests itself in so many ways: analyte loss, poor sensitivity, poor peak shape, the passivation that Udit talked about. And as you can imagine, this has really plagued us for decades. And our research teams, our application scientists have shown us just how pervasive it is. Small molecules, large molecules, QA, QC, clinical, biopharmaceutical characterization. You get the idea. It's an age-old problem, and Acuity Premier is the solution. Ian mentioned we revolutionized the industry in 2004, and here we are again with the next generation LC platform.
Very profound things. Thank you, Erin. And now, Diane, we'd love to hear about your role in this industry and what redefining separation science means to you.
Thanks, Matt. I manage our water scientific operations group. We are a team of global scientists with deep bench strength in analytical chemistry and very much in-depth application experience across all the markets we serve. We are the voice of our customer. Many of us were customers before joining Waters, and it's a role we do not take lightly. We challenge our systems and solutions each and every day to ensure that we're bringing the best, the very best in chromatographic science to the market. Like Erin, I'm a classically trained analytical chemist with decades of bench experience in environmental chemistry, crop protection science, and life sciences. We've already talked a lot about what we did back in 2004, where we looked holistically at the system and made advances in particle science and in engineering of the LC systems, the detectors, to really ensure that we could address customer needs, chromatographic needs of needing to go faster, needing to learn more about that sample. And as I fast forward to today, those fundamental needs have not changed. But what has changed is the overarching complexity of the molecules that we're looking at and understanding of human biology so that we can create, new and novel treatments. And, we've been able to learn because of UPLC, because of improvements in detector sensitivity, such as mass spectrometers. We've seen how critical these interactions can be with analytes that just love metal and love metal surfaces, and the amount of lost time and money, and frankly, just frustration, how frustrating frustrating it is, as Aaron just said, about not being able to see your sample and having to spend so much time troubleshooting. And while 2004 seems like it was a lifetime ago, you know, Waters has not stopped in its research. We've introduced, you know, new technologies, advances in the system. But today, what we're really introducing is that step change now in chromatographic science that can help to really minimize those risks, so that scientists can focus on developing and inventing new technologies and not having to worry about their system.
There's so many interesting thoughts packed into each one of those responses. Thank you, each of you. Before we go deeper still on benefits and significance, perhaps we can describe the technology some. Like with UPLC, the Acuity Premier technology is something born by both advances in instrumentation and material science. So Aaron, what is the material science here?
Thanks, Matt. And, you know, we are so proud of the work that has gone into this. It's been years in the making. You mentioned joint invention here, and I want to go back just for a minute to that birth of Acuity that Diane talked about, that Ian talked about. So that was a case where chemistry did the material science. They developed those sub-2 micron particles, and our systems colleagues developed an instrument that could deliver on that performance. And that was the birth of UPLC. So much like we did back then, this is another example of the system and the column working together and being designed for each other. The premier solutions that we're talking about today are the result of really a holistic approach to addressing this ubiquitous problem of adsorption. From plates and vials to the columns we released last year, and now sort of the end of the journey and bringing it all together with the system. It wasn't an easy problem to solve, though, to be fair, particularly when one is aiming at a very universal approach. You know, as I mentioned, this is years in the making. And, you know, we leveraged decades of experience in surface modifications, the subtleties, and developed from the ground up proprietary surfaces and indeed the accompanying manufacturing process. And, you know, the result was a family of very resilient, engineered chromatographic surfaces, which combine the attributes of fused silica and polymers, and ultimately deliver a new set of capabilities. It's very different from any of the workarounds that you see out there today. You might see folks using alternative materials of construction, additives, passivation techniques, but these all have shortcomings, and there's a compromise somewhere. What we've developed here allows us to maintain the efficiency of the UPLC hardware, but minimize those metal and other interactions. So in the case of the metal interactions, the electron-rich analytes cannot reach electron-deficient metals in the separation path. We've eliminated the need for additives, which can cause suppression or selectivity changes. And we've used materials of construction with proven mechanical and chemical stability. It's really, you know, a remarkable advancement in materials, namely hybrid inorganic and organic silica. So we should mention too, just for a moment here, that the manufacturing process is also developed from the ground up, where we subjected surfaces to exceptionally rigorous testing to prove to ourselves and our industry that we have that stability, that chemical, mechanical stability, reproducibility that our industry requires, quite frankly. You know, customers don't want to worry about how reproducible the separation is. They just want it to work out of the box. And so our global teams of engineers and of scientists, including, you know, a lot of great applications work in Diane's team, have tested thousands of components and columns, focusing on that off the bat performance. And it's really the breadth and depth of this testing, the rigorous manufacturing tests during development, and the fundamental underlying material science that gives us that confidence.
Hearing about these surfaces never gets old, even though, as you've been explaining, we've been diligently working on it for years. That said, the capabilities of the Acuity Premier is not exclusively to hybrid surfaces. So I'd like Diane, maybe, can you explain to us how there are some other factors involved in the new system?
Yeah, sure, Matt. You bring up a good point. Really what we've been talking about so far has been the sample interaction from the container that it's in, the vessel that it's in, all the way through the flow path of the LC system. So it's not only those interactions that are important, but also understanding the overall construction of the pumping system, for instance, and really ensuring that that entire system is corrosion resistant, right? You think about some of the mobile phases that are used in these applications, and they can be incredibly corrosive. And so the Acuity Premier LC system has additional components that are made from titanium and other very inert metal alloys. And this really, in combination with those hybrid chromatographic surfaces that Aaron just talked about, helps to prevent analytes from that non-specific binding and from undergoing reactions with metals across the system.
It's a beautiful example of systems engineering and a lot of technology to be talking about. If there's any question, remember that all attendees, you're welcome to be sending in questions. We're going to be here for discussions now and carrying forward for the day. Let's turn to thinking about how these advances in technology give compelling benefits. Now, this is a question for Ian. When you first got involved and came up to speed of learning the capabilities of Premier technology, what was your biggest aha type of insight?
Well, thanks for that, Matt. I guess my first thought, which was essentially a trivial one, but it's worth sharing because first thoughts are usually trivial, was, now at least I don't need to ruin a mass spectrometer by adding iron pair reagents to my mobile phase. Because there are a lot of analytes like citrate and lactate and ATP and other things like that, that in order to get any chromatography at all, particularly if you're trying to determine them at low concentrations, up until very recently, that was the only way of doing it. You used an iron pair reagent. Now, for those of you who've got a few spare mass spectrometers that you don't really need, it's still a good solution. But for me, that's a major advance because it's an instant saving of a lot of dollars. And then I saw some of the results that people were getting in waters for things like phosphate prodrugs, where the peak shape was suddenly wonderful. And secondly, the dynamic range that was obtained was a lot higher because the standard curve went all the way down to the bottom rather than stopping halfway, as it usually did when the balance between non-specific binding and chromatography shifted from chromatography to binding. And I then started thinking a bit longer, and I went back to some work I'd done as a much younger man for the pharmaceutical industry, where I was in a drug development team, where the chemists had actually very nicely designed some compounds that were actually specifically for metal chelation. So the peaks that they provided were some of the most interesting peaks I've ever seen. They went from chair shape to awful. And for us, this was a new problem, and it took us a long time to solve. We even went as far as adding iron to the mobile phase in order to try and outcompete with the column. That didn't work, don't try it. So the great thing about these columns is, if I'd had them, I wouldn't have known I had a problem because there wouldn't have been one. Our solution was to put picolinic acid in the mobile phase, and that worked a treat. And we only had to put it in at 1 millimolar. So that would have been like an iron pair reagent as far as a mass spectrometer was concerned, so not really viable. So, not having a problem sounds like my idea of a good day.
Same for me. A fantastic way to describe it. Thank you.
Yeah.
It's sounding like there's more accuracy, more precision on hand. Diane, can you expand on all the ways that our scientific teams have been unearthing these benefits?
Absolutely, Matt. As a scientist, we think about those aha moments, and they're very rare, right? It's not every day we have those moments, but when they happen, they are just, the excitement is electric, and just the change in mood and motivation can just be so tangible. And you know that our chemists at Waters and our customers are extremely demanding. They require the best of the best of their systems and solutions. And when my own chemists, our own chemists, and our customers have multiple aha moments, you know that there's something there. There's something really there with the technology. And that's what we've seen so far with Acuity Premier. There's a couple of key examples I would say that I'd like to highlight today. One is what Ian just mentioned about phosphate prodrugs. So one of the first drugs that we looked at was dexamethasone sodium phosphate. Most of you have probably heard about this. It's been used most recently to treat COVID-19 patients. And under traditional chromatographic conditions, we see significant tailing and poor sensitivity and poor calibration curves. And from the first injection, we saw an 8-fold improvement in sensitivity. And this is important for assays such as impurity assays, bioanalytical assays, where you need the ultimate in sensitivity. And there are many, many phosphate prodrugs out there. And so we really anticipate significant benefit. One of the most powerful examples, Carrie Smith worked on this. She worked on the citric acid cycle, or the TCA cycle. Those that probably are familiar with this cycle know how critical this cycle is to understanding disease mechanisms, whether it's inborn errors of metabolism, heart disease, cancer. It's so critical, foundational to our understanding of human biology. And when Carrie ran the organic acids from that cycle, she saw a 100-fold improvement in sensitivity. Let that sink in for a moment. 100-fold improvement. We essentially went from seeing what looked like a blank chromatogram, almost a flat line, to actually being able to see peaks and to be able to quantify those peaks. Think about the sorts of new discoveries about human metabolism that we can learn by being able to measure these much more accurately, and the overall treatments that may result. It's just fantastic science. And the third application are B vitamins. I think many understand the difference, very structural differences in B vitamins. And some of them do have metal chelating moieties. And, it's essential to understand B vitamins are very important for human metabolism and physiological function. And so there's a lot of testing and analyzing of these B vitamins in not only nutritional research, but also then from a food and beverage QC standpoint. And typically these analyses are single analyte methods due to the complexity of the molecule, due to the complexity of the matrix that these vitamins are in. And what we've seen to date has been a 3-fold improvement in the sensitivity for many of these B vitamins, and also the potential now to combine some of these methods for much more streamlined and reproducible analyses. So these are really just a handful of examples that we've seen so far, and we continue to see each and every day those aha moments.
That's right. And so many of these things are turning out to be a new benchmark, a new standard of measurement for the assay. And I think we're going to rightfully expect to see more and more to come. One might wonder how Premier could fundamentally change some of these decisions on how do you build a new lab? How do you think about your new instrumentation? For what it offers on these historically challenging analytes, you might be thinking, how do I future-proof? To that point, Diane, what are you thinking? Where are you seeing a good decision could be made?
Yeah, Matt, I would say, thinking to my days at the bench, there are few situations as frustrating as encountering another scientist, another colleague who says, I cannot reproduce your method. I cannot generate those same data. It is incredibly frustrating. Typically, you have spent weeks, if not months, working on developing that method, qualifying the method, validating the method, and that to hear that someone else can't reproduce that is just so frustrating. And this cuts across markets and it cuts across applications. This might be a QC release assay for a new pharmaceutical in tech transfer from development to QC. It could be a bioanalysis method for clinical trial samples that's being transferred from a pharmaceutical company to a CRO. It could be a method at a food company who is running infant formula QC analyses and they cannot reproduce that AOAC method. So you just never know when you might run across these situations, and the tangible and intangible costs can be devastating. And so what we've seen so far with Acuity Premier and the Max Peak high performance surfaces is that we now have confidence in that overall method reproducibility from system to system, from chemist to chemist, and from labs in Boston to labs in Shanghai. And I think as we think about building out our laboratories and future-proofing our space, this technology will be a critical component going forward.
It's a remarkable vision, it certainly is. And hearing and seeing these ideas about how it could be done better and remove some of the pains of the laboratory work makes me think of a well-known Confucius quote, "To know what you know and what you do not." Aaron, on that concept, what might that get you thinking about?
Well, so just to, on future proofing just for a minute there, and then on sort of the knowing what you know and knowing what you don't know. The future proofing, just to take a minute there, it distills down for me to two concepts really. And Diane alluded to a lot of it and was explicit about much of it. None of us can predict what's coming next, nor what will take priority next. We know that project priorities shift on a daily basis. Today's fringe research is tomorrow's routine. Look how quickly science has advanced over just the past few years, lightning speed. Who knew 2021 we'd have the first widely distributed genomic vaccine? I remember just a few years ago, there were 2 oligonucleotide-based drugs on the market. Now there are 10 or 11. The second concept there is around risk and how much you want to take. Diane mentioned the scariest things we face as scientists. One, that somebody can't reproduce your method. The other is to realize later that you missed something. And, in the case here, without these surfaces, we don't even realize there should have been a peak there or that the concentration we're observing is wrong. And not because the analyte wasn't in the sample, but because it got lost along the flow path. We've shown this technology to profoundly benefit a broad and diverse chemical space. You really got a great sense of that from Diane. It's about as widespread as one could imagine. And so, to me, you just can't afford to miss anything. I think about one last example there, and I believe both Ian and Diane touched on it briefly, but biomedical research, you have got to be able to see everything. You've got to be able to reproduce that data across different labs. You've got to feel confident in up and down regulation. So you simply can't afford to be wrong, or you fundamentally set research back potentially years. Getting back to that quote, Matt, one thing I think it's fair to say for sure is that we don't know what we don't know. Or in this case, more to the point, we truly did not know what we were missing.
That's right. Extraordinary when you think about the implications on everything and turning a new leaf and getting new opportunities in the lab. So we confirmed there are massive effects for metal-sensitive analytes, but I believe there's also something more holistic to it all. Having precise control of the chromatographic surfaces is a huge breakthrough. No more is there variability from metals, the supply around the world, their oxides, the metallic surface imperfections. And to that point, it's genuinely seeming like we're on to the next evolution of LC. this is a question now for our last round where I'll invite each of you to hone your thoughts and give us your sharpest response on what this means for just getting better chromatography. And allow me to go from Ian to Diane to Aaron, if you don't mind.
Okay, well, for me, this means we'll be able to dig deeper than ever before into things like the metabolome and the phosphoproteome and stuff like that. It will, as Erin said, allow us to see the things we didn't know we were missing, and that's really important. I think it'll help us to address problems that before were technically very demanding and actually really effectively impossible. So I want to steal another quote, but from something more recent, which is Star Trek. You know, it'll allow us to boldly go where no one's ever boldly gone before. Probably boldly.
Yeah.
And Diane?
Excellent. You know, as an analytical chemist, our mantra is always accuracy, precision, reproducibility. And what I see the Acuity Premier solution is bringing us more confidence in our results, in the data, the overall reproducibility of our methods. And really that scientists can now focus on new discoveries, can focus on inventing new solutions, new pharmaceutical drugs, and being able to release their products more confidently and not worrying about the method or the system. I think it's just a liberating, technology where we can now get back to focusing on the fundamental science.
And Aaron?
Yeah, Diane, Ian, I think you summed it up wonderfully. To me, this is the new standard in LC separations. No more passivation, conditioning, sensitivity, the likes of which we have never seen. The real question, and I'll make this very simple, is why wouldn't you use it? We started out getting the aim to look for the best possible chromatographic performance and solve a really tough problem. Ultimately, what we delivered was the next generation LC platform, which is the Acuity Premier solution.
Thank you, panelists. Thank you. What a well-painted picture. With that, I'd like to thank everyone for joining us today. We've passionately pursued this technology, and we've been very eager to introduce it to the analytical community. It's a rare opportunity to help evolve separation science. We look forward to catching up on how you'll be doing this with the Acuity Premier. With that, take care all, and please do not hesitate to reach out and ask some questions.